Okay na sir?
ఒక 2 minutes sir. సార్ స్టార్ట్ చేయండి సార్.
ఓకే,
స్క్రీన్ పెద్ద చేయండి.
మంచి ఆలోచన.
ఓకే, స్క్రీన్ పెద్ద, స్క్రీన్ గింతే. ఓకే సార్, ఇంత పీపీటీ ఇంతే కదా. ఓకే, ఆల్ ఆఫ్ యూ గుడ్ ఈవినింగ్. Today we are discussing about the enzyme chapter. Yesterday also. Okay. Today we are discussing about the enzymes remaining chapter. So that is the all of you discuss here mechanism of enzyme action. So mechanism of enzyme action. So in this, so all of you are observing. So here E means enzyme and the S means substrate, and the here you are observing E enzyme and S is
సార్ మీరు స్క్రీన్ పెద్ద చేయండి సార్.
సార్ ఇది స్లైడ్స్ సార్, పీపీటీ ఇంకా ఇంతే ఉంది సార్.
నా స్లైడ్స్ పెద్ద చేయండి సార్. అది వ్యూ దగ్గర కొట్టి వ్యూ దగ్గర పోయి
సార్
ఫుల్ స్క్రీన్ కొట్టండి.
ఎక్కడ ఫుల్ స్క్రీన్ అంటే
స్లైడ్స్ ఫుల్ స్క్రీన్ పెట్టండి.
నా దగ్గర ఫుల్ స్క్రీన్ ఉంది సార్, నాకు మాత్రం. సార్
ఒక్కసారి ట్రై చేయండి సార్. మళ్ళీ స్లైడ్ షో అని పెట్టేసి
స్లైడ్స్ మళ్ళీ స్టార్టింగ్ అటు మొత్తం అటు పెట్టి ఫుల్ స్క్రీన్. సార్ ఓకేనా సార్? ఓకేనా సార్? ఓకేనా సార్?
ఓకే సార్ చేయండి, స్టార్ట్ చేయండి.
Okay, right. Good evening, all of you. So today we are discussing remaining the enzymes chapter. Yesterday we discussed the enzymes introduction. So some enzymes are behave like the enzymes. All enzymes are the made up with the proteins, but all proteins are not enzymes. And we have to discuss the what is that chemical reaction. In a chemical reaction, we understood what is that. So in the formation of new substratum or the new product, so the substratums are between the substratum, some bonds are broken, and during the formation of the products, the new bonds can be formed. So that is the chemical reaction. And then after the reaction rate, P by T, the given the per unit time, how much products are formed by the reaction. So already we completed the what is that, the reaction can slow with the absence of the enzymes. In the presence of the enzymes, the reaction is very fast. So today we are discussing about the what is that mechanism of enzyme action. Here all of you understood. Here E means it is denoted enzyme, S substratum. So here you are this is the what is that enzyme. Each enzyme have to the some points, some folds or some target points. So these are the fitting points. So these are also called as active sites. These active site is so help into the what is that, the fixation of the substrate at the point of active site. So here this is the substratum, these are the enzymes. These substrates are fixed in the what is that, enzymes. Here you are clear, observe that. That's time we have to the what is, we can get the what is that, enzyme substrate complex, E and S substrate complex. Then after we can get the enzyme product. So here you are observing these enzyme is react on the substrate, and the red color you are observing these enzyme reaction. After reaction, so enzyme color is changed, and the here enzyme is the same. So these substratum, so color here changed, you are observing. So these red color. So what is that here? Understood what is that enzyme and substratum? We can get the what is that enzyme substrate complex. Then after the enzyme is react on the what is that substrates. So and formed the what is that the substratum is modified into what is that as a products. So here you are of the enzyme and the P. So these are the what is that enzyme action, a mechanism of enzyme action. Here first point is what is that each enzyme have to the some spaces for the fitting of the substrate. That fitting part is called as active site. So in the active site, the substratum can be fixed. That's time we can get the what is that enzyme substrate complex. After that, as the enzyme react on the substratum can convert into the what is that products. So these yeah the after that the substratum is converted into the product. The enzyme is normal. So these are called as what is that mechanism of enzyme. In examination gives to the shows the identify the which statement is right given to the so you are observing here sometime given to the E plus P and EP and the ES E plus S this type of reaction given opposite type but we are the correct answer is what is that enzyme and the plus substratum and the enzyme substrate complex and the enzyme and product. So then after here you are observing enzyme and product here substratums are formed and convert into the what is that products. So let us now we discuss the so lock and key hypothesis. This is the proposed by the Emil Fischer scientist. So lock and key hypothesis. You are observing here. So clear that this is the substrate. Okay. So here this substrate is the what is that lock like structure. So like lock and the key is what is that enzyme you are observing. Substrate is what is that lock and the key is what is that enzyme. Yeah, this is the so here the key is what is that enzyme. So the substratums are fixed in the what is that key. So here the key and the lock. So these are the what is that enzyme and the substrate. So now the key is inserted into the what is that lock. So means substrate in the place of the enzyme active sites. You are observing these enzyme active sites are fixed into the what is that in the fit into the substrate are fitted into the enzyme active site. That's time we can get the what is that enzyme substrate complex ES complex. So that is you are observing ES complex. So here you are observing enzyme and the here substrate. So these are the active sites and these black color are the what is that is inserted into the enzyme substrate. These substrates are inserted into the enzyme at the where in the what is that active site port. So these are called as enzyme substrate complex. Then after the enzyme is react onto the substratum and formed the what is that product. Here we compare the imagine with the what is that substratum as a lock and the enzyme is a key. So here you are observing these theory can explain about the enzyme enzyme specificity means the each enzyme can shows the specificity means for example urease enzyme. The enzyme can be react on the only urea, but it is not a glucose on the it is not a work or it is not a react on the glucose. Similarly, the specific key is react with the specific lock. So that's time the lock can be open. The lock open is called as product. So here what is that the enzyme the lock and key hypothesis can explain about the what is that. So lock and key hypothesis explain of the enzyme specificity. So this is the proposed by the Emil Fischer. So let us now induced feed hypothesis. It is proposed to the Koshland scientist. So here you are observing this is the enzyme and the substrate. So these substrates are fitted into the enzymes at the active sites in the where in a active site places and the former the what is that enzyme and substrate complex. But in these enzyme is react on the substrate and form out the product. But this theory is utterly failure theory. Why? Because this theory is tells us it is not explain about the enzyme specificity. So here, like for example, urease enzyme is there. Urease enzyme is specific, shows the specificity. Generally, it shows the specificity. It can be react onto the what is that urea. According to the induced fit hypothesis, the urease enzyme can be react with the urea and the glucose or starch. So it is the what is that? It is not a possible. So according to this theory is follow the what is that? The enzyme specificity is not followed. So that's why it is the failure theory. Urease enzyme is urease enzyme can react on the urea, but it is not react onto the so another one substance. But in a induced fit hypothesis, explain the what is that? The urease, urea, not for only urease, any one enzyme. The any one enzyme is react any substrate. But according to this theory is not a it is not a correct. So that's why it is the failure theory. So here you are observing. So these according to lock and key hypothesis we can explain. And what is the in a NEET examination, just one who proposed Emil Fisher, and the lock and key hypothesis comparison. So lock is compared to what is that? Substratum. Key is the what is that? Enzymes. So what is the lock and key hypothesis? Explain about the only 2 points important. So what is that? Enzyme specificity. And the next one, induced fit hypothesis. It is proposed the which scientist? Coastland scientist. So that is important. And it cannot be explained about the enzyme specificity according to these the hypothesis. So what is according to these hypothesis? Only you are observing first one, clear that the enzyme can react any substance. It cannot be react on specific substrate. So that's why it is the
Sir.
failure.
సార్ సైడ్స్ లో అయితే వస్తలేవట సార్, అది స్లైడ్స్ షో అయితే లేవట.
ఫ్లో అవుతులేవా?
స్లో మీరు చూపిస్తలేరు కదా స్లైడ్
చూపిస్తున్నా సార్.
ఇక్కడికి వస్తలేదు సార్, చూడండి ఒకసారి మార్చండి.
చూడండి ఇప్పుడు ఇగో చూడండి, ఫ్యాక్టర్ అఫెక్టింగ్ ఎంజైమ్ చూడండి.
నీట్ పీ కుమార్ సిద్దిపేట అని వస్తుంది సార్, అంతే ఏం మారలే.
అయ్యో స్క్రీన్ షేర్ చేసినా సార్,
ఇక్కడ రావట్లేదు సార్.
మరి ఇక్కడ చూడండి, స్క్రీన్ షేర్ చేసిన నేను సార్, ఇక్కడ స్క్రీన్ షేర్ చేసిన
మీ ఇక్కడ చూపిస్తే సార్, మన జూమ్ లైవ్ లో కూడా చూస్తున్నా నేను.
చూడు మళ్ళీ ఒకసారి చూడండి. ఇప్పుడు నేను ఇప్పుడు కనిపిస్తుందా సార్? మీరు
కనబడుతున్నారు ఇప్పుడు.
ఓకే, ఇప్పుడు ఒక్క నిమిషం ఆగండి. ఇప్పుడు కనిపించిందా సార్?
ఇప్పుడు ఏది మీ పేరు అది కనబడుతుంది అంతే.
ఓకే, నెక్స్ట్ అట్లా చూద్దాం. ఇప్పుడు సార్
ఏం రాలే,
ఫ్యాక్టర్ అఫెక్టింగ్
ఏం రాలే.
అగో ఇక్కడ నాకు కనిపిస్తుంది సార్, ఇక్కడ వస్తుంది సార్. ఇగో ఇప్పుడు
రావట్లేదు సార్.
టెంపరేచర్, లో
టెంపరేచర్ ఏం లేదు, మీ పేరే సిద్దిపేట వస్తుంది అంతే.
అట్లెట్లా సార్? నాకు ఇక్కడ కనిపిస్తుంది సార్.
స్క్రీన్ షేర్ అవుతుందా?
అవుతుంది.
నెక్స్ట్ స్లైడ్ మీద క్లిక్ చేయండి.
ఇక చూడండి ఇప్పుడు
సార్, నెక్స్ట్ స్లైడ్ మీద క్లిక్ చేయండి సార్. ఫస్ట్ స్లైడే కనబడుతుంది.
ఫస్ట్ స్లైడే కనిపిస్తుంది అంట. ఇక్కడ అవుతుంది సార్. మరి ఇప్పుడు
సార్ ఇప్పుడు స్క్రీన్ షేర్ ఉంది కదా సార్, దాన్ని క్లిక్ చేసి సెకండ్ ఉందా, కనబడుతుందా సెకండ్ సార్?
కనిపిస్తుంది, నాకు ఇక్కడ కనిపిస్తుంది సార్.
సార్ ఫస్ట్ మీ ఉంది కదా మీ పేరు, సెకండ్ స్లైడ్ మీద క్లిక్ చేయండి.
స్లైడ్ కనిపిస్తుంది, ఫస్ట్ ఒకటే కనిపిస్తుంది అంట. మళ్ళీ స్టార్టింగ్ సార్. ఇప్పుడు సార్ మీకు ఇంకేది కనిపిస్తలేదా? ఇది ఇప్పుడు చూడండి సార్.
ఇప్పుడు అయింది. ఓకేనా సార్?
ఓకేనా సార్? ఇప్పుడు ఓకే సార్, రైట్. సో okay sir, now lock and key hypothesis, it is proposed by Emil Fisher. So here lock compared to the substratum and it is the key. and here the enzyme, the key is, so have to the specific active sites, it can be fixed into the substrate. So then after the specific key can react with the specific log. So similarly, the enzyme can react on the specific what is the specific substratum. For example, urease. So that enzyme is react on the urea. So this is the lock and key theory. And the next one you are observing, induced feed theory. So induced feed theory, now here it is proposed by the Coastland scientist. So here, according to this theory, the enzyme is react, the enzyme can react on the any substrate. So but it is not a correct. So that's why it is the failure theory. According to this theory, the urease enzyme can react on the urea and another one substrate. So that's why it is the failure theory. So dear students, you are observing. So first you are observing lock and key hypothesis can explain the enzyme specificity. And here your enzyme, this is the substratum fixed in the active site, and the what is that substrate molecules can form as a product. So what is that lock and key hypothesis is, what is that? Emil Fischer. So next one, induced feed hypothesis. So according to induced hypothesis, next you are observing. So lock and key theories gives to the information, what gives the shows the what is that enzyme specificity, and the induced hypothesis is what is that failure theory. It is not shows the enzyme specificity. Next, factors affecting enzyme activity. Here you are observing. So first one, the factors are, the number of factors are affected on the enzyme activity. So in this, so first one, the number of factors are there. So number one is the what is the temperature. So in a temperature, first one, low temperature. So one is the what is that low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroy the enzymatic activity. So because of it have to the this range is generally the enzyme activity is 25 degrees centigrade to 30 degrees centigrade. So above the 25 degree to 30 degrees centigrade, so the enzyme can enzymes are destroyed, and the enzymatic activity is the decreased. So what is that? The temperature, it is the enzyme activity is range is what is the 25 degrees centigrade to 30 degrees centigrade. And the next one you are observing pH. So it is the best work in a neutral. Different kinds of the enzymes shows the different pH values. Here you are observing the increasing the pH value, the rate of the reaction also increasing. So here this is a pH and rate of, for example, pH is increasing, so automatically the rate of reaction also increasing. So for example, amylase, urease, catalase. So these enzymes pH value is 7.0. Here you are observing the pH value is increasing, automatically the reaction can be increased. So next one. So here concentration of substrate, here clear that. So here now you are observing, if here this diagram x-axis and y-axis your the Vmax velocity. So if increase substrate concentration, the velocity of enzyme reaction rises at first. First one, what is that? Enzyme reaction is rises first, and the reaction ultimately reaches a maximum velocity. You are observing from these beginning to increasing the what is that reaction velocity. So increasing, so it reaches to the what is that, these are the what is that maximum velocity. Clear that maximum velocity. Then after you are observing substrate concentration required to cause the half of the maximum reaction rate is termed as Michaelis Menten. Km, it is the very, very important. Only this is the in NEET purpose, only this. So total this chapter in a NEET exam point of view, only one bit the chance may or may not be. So only one bit is important from these. Only completed the MGM scientist names, and the next one is the lock and key hypothesis scientist names important, and the what is the use of theory. So the next one Michaeli maintain constant. So what is that? This is the you are half of the rate. You are observing half of the rate. This place you are half of the plate. So substrate concentration required to cause half the maximum reaction of the maximum reaction rate is termed as Michaelis Menten constant. So represent approximate inverse measures. So next concentrate here the what is that? This is the very important way. Only this is Michaelis maintaining. So Michaelis maintaining. So next one you are observing enzyme inhibition. So what is that meaning? Inhibition. Inhibition means what is that? Shuts off the enzyme activity. Simply we are saying what is that? Shuts off the enzyme activity. The process is called as inhibition. So when the binding of the chemical shuts off enzyme activity, so that is called that process is called as inhibition. So here inhibitors are three types. One is the competitive inhibitor. Second one is the non-competitive inhibitor. So first one competitive, second one is the non-competitive, and third one is the allosteric feedback inhibitors. So first, dear students, so you understood the and remember it the what is the meaning of the inhibition. So that is the inhibitor and inhibition. So shut off the enzyme activity process is called as inhibition. So then after you clear that, so inhibition next inhibitors are three types. One is the competitive inhibitor, and the second one is the non-competitive inhibitor, and third one is the allosteric and feedback inhibitors. Okay, then after first one, what is that? Competitive inhibitor. Simple logic point in NEET point of view. Already you completed the IP, so that's why only you are observing the total concept. So what is that? Competitive inhibitor. So here the enzyme, the substances are closely resemble to the substrates and the inhibit enzyme activity. So like for example, you are observing. So this point is very important. Substances closely resembles to another substrates and the inhibit the enzyme activity. For example, you are observing inhibition of succinic dehydrogenase by Malonates. So here similar dehydrogenase is the one structure. Succinic dehydrogenase is the one structure. It is the Malonates are similar to the succinic dehydrogenase. So here you are observing the succinic dehydrogenase is similar. It is react with the what is that? Succinic acid generally. So but the succinic acid is the malonate is also similar or resemble to the succinic acid. That's time instead of succinic acid, the malonate is fixed at the active site in the succinic dehydrogenase enzyme place. So that's time these are formed the what is that? Enzyme and substrate complexes. Enzyme and the substrate complexes. But the substrate is not converted into the products. So that is the very important. So similar, you are observing what is that? Substances are closely resembles to substrates and it is the inhibit enzyme activity. Clear that? So example is what is that? Succinic dehydrogenase. Already you know what is that? The succinic dehydrogenase can react on the succinic acid. It is a common method. But here the malonic acid also similar to the succinic acid. in this situation, so in actually the succinic dehydrogenase is react on the succinic acid. But here, instead of the succinic acid, the malonic acid is reaches to the active site of the succinic dehydrogenase places. But both are the formed here also enzyme and the substrate complexes. Okay, but the malonic acid is not converted into the product. So these are called as competitive inhibitors. So in examination, the given to the what is that? The succinic dehydrogenase best example for competitive inhibitor, malonate, succinic acid malonates are. So what is that? Competitive inhibitors or non-competitive inhibitor. So allosteric one. So these type of very, very important. You are prepare these manner only. Competitive means resemble the substrates. So example is what is that? Succinic dehydrogenase. So that is the clear. Next, non-competitive. So non-competitive also these are the both are not similar and formed the what is that? They can form the and products. In a competitive inhibitors, we cannot get the products, but is there non-competitive also? They cannot reach us to the different kinds of the what is that? Products are not formed. Means what is that? Here non-competitive you are observing. For example, this is the competitive. Here what is the succinic acid and malonic acid. In a non-competitive, these are the substrate can not fixed at the active site. They can fix it another place. That's time enzyme what is that? Substrate complex are formed, but they are also not gives to the products. So only different is you are observing competitive inhibitor, non-competitive inhibitors means in a competitive inhibitors, these are the fixed at the what is that active site place. In a non-competitive means they are not the fitted at the active site place. Any places, enzyme, any another places they can fit it. But the that's during that time the enzyme cannot react on that. So that is the non-competitive inhibitor. And next last one is the what is that? So feedback inhibitor. So in this feedback inhibitor, you are clear that the end product of you are clear that the end product of a chain of enzyme catalyzed what is that? Catalyzed reactions inhibits the enzyme of the first reaction as part of homeostatic control of the metabolism. For example, you are observing A to B, B to C and D and gives to E. So in the from the D again the reaction is comes reversely. So this is the homeostatic control method. So here when you are observing glucose is converted in the presence of glucose consumed the ATP and in the presence of exokinase enzyme gives to the what is the glucose 6-phosphate. So and next this glucose 6-phosphate cannot form to the glucose. That's time the exokinase can be act as a what is that? Inhibit. So similarly this process is very different. So now we have to the different kinds you are observing. The end product of a chain of enzyme catalyzed reaction inhibits what is that? The enzyme of the first reactions as part of homeostatic control of the metabolism. So best example is what is that? Glucose, and the gives rise to the what is that? Glucose 6-phosphate, and these glucose 6-phosphate is inhibit the exokinase enzyme. So here today class you are observing important points. All of you observe that mechanism of enzyme action. So in this what is that? Enzyme and the substrate. So both are the form of the what is that? Enzyme substrate complex. And then after what is that? Enzymes substrate can formed as a what is that? Products. So here reversely your enzyme is normal. and the substrate can the form the what is the products. So these are called as what is that mechanism of enzyme action. So next one, mechanism of enzyme means what is that? The each enzyme have to the specific active site. So these sites can be help to the fitted of the substratum, and the enzyme can react on the substrate at the active site. And when both are the fused, that complex is called as enzyme substrate complex. So the substrates can converted into the what is that products. So this is the mechanism of enzyme action. Next, lock and key hypothesis, very important. The scientist name is Emil Fischer. So Emil Fischer, here the what is that enzyme is the key, and the lock is the substrate. So the it can be the a specific key can react on the specific lock. That's time the lock can be open. So for example, our house key cannot be fitted on the another house lock. So that's why, so the specific key can be react on the specific lock, and that can be open. Open means what is that product. Here also similar. So the for example, urease enzyme is there. This is the urease enzyme can react on the only urea. Okay. So that's why lock and key hypothesis can explain what is that lock and key hypothesis explain the about the enzyme specificity. And the next one, what is that? Induced fit hypothesis. Induced fit hypothesis, the theory is called as what is that Coastland theory also. So it is not a explain about the enzyme specificity. According to this theory or hypothesis, the any enzyme, the each enzyme can be react on the different kinds of the substrates, but actually it is not a true or correct. So that's why it is the failure theory. So according to this theory, imagine that what is that the urease enzyme can react on the what is that urea actually, but it can be react on the glucose, sucrose, any another one substrate also. So that's why it is the failure theory. And the next, factors affecting enzymes. The factors also there. The factors are generally what is the temperature and the pH. So here your temperature, the in the during low temperature preserve the enzymes in a temporarily inactive state, whereas in a high temperature also enzymes are destroyed. So the proteins are denatured by the heat. Generally the enzyme activity suitable or the temperature is, so what is that 25 degrees centigrade to 30 degree centigrade. Next one here, pH. So best work in the neutral. So the enzymes are generally work in the neutral. So that is the what is that 7. Okay. So some enzymes are shows the different kinds of the pH values. So for example, amylase enzyme and urease enzyme and catalase, these enzymes are, so in the what is that neutral, 7.0 means neutral. So increasing the enzyme pH value, automatically their activity also increasing. So that is the very important. So temperature also, the temperature also from low temperature to high temperature, slowly the enzyme activity can be increased. So after they can reaches to the maximum, above the maximum reaches, the enzymes cannot be active, means the enzymes can be destroyed. Okay. So next one, what is the pH? So what is that? The pH is the generally the all enzymes are work in a best work in the neutral. So what is that pH value is so amylase, urease, catalase is 7.0. And the here concentration of substrate. Here you are observing. So first, if we increase the substrate concentration, the velocity of enzyme reaction, what is that rises at first, and the reaction ultimately reaches to the maximum velocity. Here substrate concentration you are observing. This is the substrate concentration. It is the Km. So this is the Vmax. So this is the rate. So here you are observing the substrate concentration required to cause to half of the, you are observing. This is the slowly increasing here. So rises the first one enzyme reaction rises it first, and the reaction ultimately reaches the, this is the maximum. And then after you are observing, so the substrate concentration, the substrate concentration required to cause of the maximum of the, what is that, maximum reaction rate is termed as Michaelis Menten. So Michaelis Menten constant Km. So it is the represent approximate inverse measures. So next here, what is that, enzyme inhibition? So when the binding of the chemical shuts off enzyme activity, so the process is called as, what is that, inhibition. Inhibition is how many types? Three types. Inhibitors are three types. One is the competitive inhibitor, and second one is the non-competitive inhibitor. So third one is the, what is that, allosteric or feedback inhibitor. So competitive, already we know what is that, both are, some substrates are similar to both are similar. So non-competitive means they are the not suitable or not resemble. So here you are observing, so clear that substrates closely resemble to what is the, closely resembles to substrates and inhibit enzyme activity. So inhibition of succinic dehydrogenase by Malonates. So here enzyme you are observing, instead of succinic acid, so the malonic acid is inserted into the, what is the, enzyme active site. But here you are observing the enzyme and the substrate complex are formed, but the malonate cannot converted as a, what is that, products. But what is that, similar, both are the similar. So that's why these are called as competitive inhibitors. In a non-competitive inhibitor, so that is the very important. In a non-competitive inhibitors, you are observing the substrates are not fitted at the what is that, active site. They can fitted another, these place or these place, any another places except active site place. That's time enzyme and the substrate complex can be formed, but the substrate cannot be formed as a product. So next one, feedback inhibition. The end product of the chain of enzymes catalyze a reaction, inhibit the enzyme of the first reaction as part of homeostatic control of metabolism. So here first A to B and B to C and the D and E. Then after during the D, so again comes to back feedback and the first reaction cannot be inhibited. So here similarly you are observing glucose to glucose 6-phosphate, number of reactions are there. Glucose is the A, glucose 6-phosphate is the D. Between the B and C, between the number of reactions also there. Enzyme is similar, hexokinase enzyme, but it inhibit the, what is that, formation of, it inhibit the again formation the A. It is here glucose 6-phosphate is not converted into the glucose. So what is the, why they are not formed, why the glucose 6-phosphate is not converted into glucose means here the hexokinase act as a inhibits. Okay, now we discuss the some bits. So who first used the term enzyme? So already yesterday we completed that. So what is that? Who first used the term enzyme? The enzyme term coined by the which scientist? Kune. So which scientist? Kune. So that is the J.V. Sumner is not there. So first correct answer is, what is that? Kune. And the next, who coined the term gymase for enzymes in yeast? So gymase. So Edward Buchner. So which scientist? What is that? Edward Buchner. So that is the important, Edward Buchner. Next, who coined the term gymase for enzymes in yeast? Edward Buchner. And the enzymes are basically, or all enzymes are contained. What is that? All enzymes are, what is that? Proteins. What is that? Sugars are not right answer, and the fats are not answer. What is that? Vitamins. So here clear that all of you are observing. So enzymes are basically, or the all enzymes are contain the what is that proteins. All enzymes are made up with the proteins, but all proteins are not a enzymes. So that is the very important. So here answer is what is that proteins? What is that proteins? Next here enzymes are proteins. It was suggested by, so which scientist is suggested? So what is that enzymes are proteins? So that is it was suggested by. So what is that? Miller, Sumner, Patcher, and Leeuwenhoek. So here you are observing Sumner scientist. Sumner. So that is a very important. Sumner scientist is stated that or suggested that enzymes are proteins. Enzymes are proteins. It was suggested by the which scientist? And the Sumner scientist. What is that? Sumner. And the who got the Nobel Prize working on enzymes in the year 1978. Arbut and Nathans. So Nobel Prize working on the enzymes. So they found the actually Nathan scientist we can study it in the molecular biology. So molecular seizures, endonuclease. So restriction enzymes, the scientist is give the name, they are called as molecular seizures. So what is that? Answer is who got the Nobel Prize working on the enzymes in the year 1978. So V. Arbutter and the D. Nathans. So that is the very important. Next, name the scientist who for the first time isolated and purified the enzyme urease in the crystalline form, jog bean. So scientist is this year is very, very important. Kuhn is 1826. This the Sumner 1926. Sumner is the what is that? 1926. Which of the following is called biological middleman? So these are which of the following is called as biological middleman? Important. So here first hormone and the vitamin and the enzyme. Okay, 2 minutes, sir. 2 minutes. Okay, all the above. So here is what is that enzymes. So next you are observing the lock and key model of enzyme action illustrates that a particular enzyme molecule. Here answer you are the lock and key. So just now we are completed this lock and key model enzyme action. So here you are observing may be destroyed and the resynthesized several times, interact with the specific type of substrate molecules. It is the interact with a specific type of substrate molecules. So that is the answer, the lock and key model. And the enzymes were discovered for the first time in, so it is the important yeast. So you are discovered that enzymes is the discovered. Enzymes were discovered for the first time in yeast. And the who discovered the coenzymes? It is the very, very important coenzymes. Tomorrow we will, next class we can discuss about the coenzymes. So the coenzymes are Meyerhoff. What is that? Sorry, Fritz Lippmann. Fritz Lippmann. Next, a competitive inhibitor of succinic dehydrogenase is a. So what is that? Malonate. What is that? Malonate. Example is what is that? Malonate. So in a plant's enzymes are present. What is that? So only in flower, only in leaves, in all the living cells of plant body. So this is the C is the answer, in all the living cells of plant body. Okay, sir. Sir. Sir.